Prior to the start of mitosis, interphase occurs. During this stage, cellular organelles double and DNA replicates. The nucleus and the nuclear envelope are distinct, and the chromosomes are in thread-like chromatin not yet visible. Prophase, the first step in mitosis. The chromosomes begin to condense and the nucleus disappears. As the nuclear envelope begins to break apart, the centrioles begin to move to opposite poles. Once at separate poles, microtubes begin to dissolve and around the centromeres, extending. Metaphase, the second step in mitosis. Paired chromatids align along the cell equator by the mitotic spindle. At this stage, the chromosomes are in their most condensed form. Anaphase, the third step in mitosis. The sister chromatids are pulled apart and moved to opposite poles of the cell. They are pulled apart as motor proteins move along the spindle fibers. They drag them to opposite poles. Telophase, the final step of mitosis. There are two clusters of chromosomes decondensed. Patches of new membrane fuse to form a new nuclear envelope. Mitosis is now over. Telophase is followed by cytokinesis, the division of the cytoplasm. This process occurs differently in plants and animals. In plant cells, a cell plate forms along the equator of the parent cell. Then, a new plasma membrane and cell wall form along each side of the cell plate. In animal cells, the plasma membrane of the parent cell goes inward along the equator until two daughter cells are formed. Before entering meiosis I, a cell must undergo interphase. The process is the same as it is for mitosis. Prophase I, the first stage of meiosis I. During this stage, the chromosomes begin to pair up rather than just condense like in mitosis. Each chromosome aligns with its homoglobe partner so they match up at corresponding spots at their full length. Metaphase I, the second stage of meiosis. During this stage, the homoglobe pairs line up at the metaphase plate and move towards one pole. Anaphase I, the third stage of meiosis I. During this stage, the homoglobes are pulled apart and moved to opposite ends of the cell. The sister chromatids of each chromosome are still together and do not come apart. Telophase I, the final stage of meiosis I. During this stage, the chromosomes are at opposite poles of the cell. Cytokinesis typically occurs at the same time as telophase I, forming two haploid daughter cells. Meiosis. The cells that enter meiosis II are the ones that were created during meiosis I. These cells are haploid. This means they just have one chromosome from each homoglobe pair. Their chromosomes still consist of two sister chromatids. In meiosis II, the sister chromatids separate, making haploid cells with non-duplicated chromosomes. Prophase II, the first stage of meiosis II. As chromosomes condense and the nuclear envelope begins to break down. The centrosomes move apart, spindles form between them, and the spindle microtubules begin to attach to chromosomes. The two sister chromatids of each chromosome are attached from opposite spindle fibers. Metaphase, the second stage of meiosis. During this stage, the chromosomes line up individually along the metaphase plate. Anaphase, the third stage of meiosis. During this stage, the sister chromatids separate and are pulled towards the opposite poles of the cell. Telophase, the final step of meiosis. During this stage, the nuclear membranes form around each set of chromosomes. The chromosomes decondense. After telophase, cytokinesis splits the chromosomes and creates two daughter cells. This creates four haploid cells where each chromosome has one chromatid. In humans, the result of this process are sperm or eggs. So, what is the difference between mitosis and meiosis? Meiosis has two rounds of genetic separation and cellular division. Mitosis only has one of each. In meiosis, homologous chromosomes are separate, leading to daughter cells that are not genetically identical. In mitosis, the daughter cells are identical to the parent as well as to each other.